Hello and welcome to this video summary of a research article entitled Neutral Evolution of snoRNA Host Gene Long Non-Coding RNA Affects Cell Fate Control. My name is Matteo Vietri Rudan and I am the first author of this study, which has been possible thanks to multiple collaborations in the laboratory of Fiona. We were interested in the fundamental question of how the genome can acquire new functionality over the course of evolution and whether sequences that have not been broadly conserved can contribute to this process. We focused our attention on a group of genes called small nucleolar RNA host genes, or SNHGs. These genes contain small nucleolar RNA, or snoRNA, in their introns and produce transcripts that do not encode proteins belonging to a class of molecules called long non-coding RNA, here lncRNA. Like other long non-coding RNA, SNHGs acquire new mutations relatively quickly during evolution, but they are unusually highly expressed because their transcription is required to produce the intronic snoRNAs. We use the epidermis as a model system to study the effect of SNHGs on cell fate decisions. The epidermis is a multi-layered epithelium made up of keratinocytes. The basal layer host stem cells that can either self-renew or begin a process of terminal differentiation whereby they migrate upwards and form the structures necessary to build the outer barrier of the body, ultimately shedding from the surface. A correct balance between the self-renewal and differentiation of keratinocytes is essential for the correct maintenance of the tissue. We found that several SNHGs are expressed in the epidermis and that their levels are dysregulated in inflammatory conditions where this equilibrium between self-renewal and differentiation is altered. When we removed these SNHGs from primary keratinocytes in culture, the ability of the cells to self-renew was reduced. We then focused on one SNHG, SNHG7, to better understand its effect on the biology of the keratinocytes. Indeed, when we removed SNHG7, the keratinocytes reduced their self-renewal and increased their differentiation, whereas increased levels of SNHG7 long non-coding RNA had the opposite effect and were able to rescue its deficiency. Importantly, this effect was not dependent on the snoRNA hosted by SNHG7. To understand the evolution of the activity of SNHG7, we studied its effects in keratinocytes from the South American night monkey, where SNHG7 is present but its sequence is different from the human. Removal of SNHG7 from these cells did not have any apparent consequence for their cell fate. However, when we expressed human SNHG7 in the monkey keratinocytes, we could cause a moderate increase in their ability to self-renew. By performing RNA sequencing and targeted mutagenesis, we were able to identify a binding site for a microRNA, miR34-5P, as an essential element for SNHG7 biological activity. Overall, our findings are consistent with a model in which abundant non-coding transcripts that are evolving neutrally can act as platforms to acquire new biological activity, even over short evolutionary timescales. These results open the door to further investigations aimed at understanding the extent to which these mechanisms contribute to genome evolution and biological diversification. Thank you for watching, and we hope you will enjoy reading our paper.